Okay, so I guess we'll go ahead and get started. A very, very early good morning to all of you, and thanks for joining the session this morning. My name is Betty Wu. I'm Vice President for Cell, Gene, and Advanced Therapies at Thermo Fisher Scientific. I will be your moderator for the session today. I have a wonderfully distinguished panel joining. Let me do a little bit of an introduction. So starting on your far right, Lakshmipathy, who is Senior Director for Research and Development at Patheon Services, part of Thermo Fisher. Beside her is Saba Ghassemi, who is, I have to refer to my notes, sorry, Research Assistant Professor for Pathology and Laboratory Medicine at the University of Pennsylvania. Karen Walker is beside her. She is Chief Technology Officer at Kyverna Therapeutics. And finally, we have Celia Adelson, who is Executive Director at the Eli and Edith Broad Center of Regenerative Medicine and Stem Cell Research at UCLA. Did I get that right?
That's a mouthful. I'm impressed.
Okay, we're going to be touching on two themes for our panel today, both contributing to faster patient access to cell therapies. The first theme is going to be process optimization, and the second theme is going to be expedited translation of manufacturing processes and technologies into clinical and eventually commercial manufacturing. So a recent survey of experts from almost 150 cell and gene therapy companies identified process optimization as one of their leading concerns and issues. And with that, they also highlighted the benefits of process optimization that range from reduction of COGS to increased quality, to increase the speed of manufacturing, as well as some regulatory compliance benefits. Now, we recognize that academic research focuses more on biological mechanisms and potential therapeutic applications for cell therapies. However, process development really geared toward manufacturing as a longer-term goal generally receives less focus. This is largely due to specialized GMP expertise, maybe facilities and resources that are tied to translating this to the clinic. That said, there is a growing trend of academic centers establishing dedicated cell therapy manufacturing facilities on their premises and collaborating with industry in order to develop more robust processes that will eventually translate into viable clinical processes. One example is the cell therapy facility that we opened in partnership with UCSF. It is a cell therapy facility opened last year to accelerate the development of breakthrough therapies. I'm sure that both Saba as well as Celia will comment a bit more on their investments at their respective institutions there. So Thermo Fisher continues to focus on supporting the development of cell and gene therapies from really discovery all the way up through manufacturing and commercialization. We do this in the form of tools, technologies, and specific process expertise. This expertise, of course, extends to our CRO and CDMO services that go under the name of PPD and Patheon. Our commitment to academic and industry collaboration is really exemplified, I'd say, through our investments. And I'm really happy to announce the launch of a new customer collaboration center that we're going to be launching in the early part of 2025. And this will allow our customers and our collaborators to have early access to our CTS products and technologies, as well as the process expertise that I talked about. So with that, let me roll a quick video, if I may, to give you a little bit more insight on the center. Okay, so hopefully that woke you all up. I told them to play it really loud. Okay, so with that, maybe I'll kick off the panel discussion here. So maybe I'll start with Karen. So I thought it was really nice that Tim Hunt had congratulated Kyverna on the successful IPO that you had earlier this year, so I congratulate you as well on that. Kyverna, as everybody knows, has been a pioneer in CAR T cell therapies for autoimmune disease. You published earlier in the year on a 3-day process that has whole blood as a starting material. I'm wondering if you can maybe kick us off by outlining a bit of the process itself and perhaps some of the challenges that you encountered along the way in developing this accelerated process.
Yeah, sure. So we have a traditional anti-CD19 CAR autologous T-cell process in the clinic. It's the traditional 7 to 8 day process, and it's oversized, clunky, expensive, all those things. So we set about designing a process that would really help ease the patient experience and bring down cost, which is investors' big concern, and make the whole experience more palatable for everyone and provide much greater access for patients. And the two things that we really wanted to hone in on were getting rid of the leukapheresis process, which is a bottleneck, expensive, and for patients with autoimmune disease, not really enticing, and a barrier to entry, frankly. And so by taking a normal blood draw and isolating the T cells and using some of Thermo's technology to simultaneously isolate and activate the T cells really shortens the process. We picked the duration to optimize for the viral vector integration. Of course, if we look at other forms of editing down the line, maybe we will be able to optimize that further. And then with the shorter process, as many other companies have published, we get a better phenotype of the cells. We are able to reduce the dose, and we are able to deliver a smaller volume back to the patients, which opens a lot of opportunity to look at changing the supply chain, maybe not cryopreserving the final product, looking at a very distributed model, not point of care. We can have that discussion later. Mm-hmm. But really serving the patients in their local community. And so that's what we aim for. We're in partnership with Elevate Bio to help us make this a reality. And we're really excited to bring it to the clinic.
Awesome. Well, continued good luck on that. Saba, you and your colleagues at the University of Pennsylvania have also developed an abbreviated process. This is also for the production of CAR T therapies. Can you tell us a little bit about your journey? So probably 5, maybe 10 years ago, you embarked on the process. You developed a process that was actually measured in weeks. You then went on to optimize that. The process was reduced to days. And I think with your latest, you now reported on a 24-hour process. So again, wonderful progress. Love to see that continued abbreviation. So tell us a little bit about that.
Sure. So I think same as anyone else, we also wanted to look at the whole process and think about new development in terms of how can we optimize the whole process. And I think the thought behind that was mostly, looking at the quality of CAR T cells. Because we know that, the longer that we keep cells outside ex vivo, the longer that they get proliferated, they lose quality. So I think the rationale behind the whole process was that we need a few good quality CAR T cells rather than a large number of mediocre CAR T cells and with lower. So we shouldn't look at the quantity, but we should focus on the quality. Well, it comes with a lot of other benefits that we cut the cost of goods, We, shorten the whole manufacturing process. We don't need the space, right? So we had the standard process, which is 9 to 14 days, that we know that Kymriah product was based on that. And we've done a lot of research and optimization to see how can we shorten this process. And then we brought it to 5 days, and then we brought it to 3 days. And based on this 3-day manufacturing, we had a clinical trial last year, which is concluded right now. And this clinical trial, we had 80% response in our patients that they were refractory or relapsed to prior CAR therapy. So we only admitted patients that they had CAR therapy before, whether in clinical trial or the commercialized one, and they must have failed in those. And now we have 80% response rate, which was such a success for us. And based on that, we are opening a few other clinical trials based on 3-day manufacturing process, and also didn't want to just stay with the 3 days. We thought why not making it shorter, providing that we can have gene integration. So now it actually paved. I think the success of the clinical trial gave us great confidence, and it paved the way to start to work on a process that right now is 24 hours. So just having it 1 day, and we have a lot of other optimization and other changes into the process, but this is our goal and we're hoping to open a clinical trial next year. And as Karen mentioned, I think the overarching goal behind the whole process, shortening the process, is that we move away from decentralized, move away from centralized manufacturing towards a decentralized. And again, I would say the same. Maybe we don't start with point of care, but more like a regional, or having more access to such a life-saving therapy that a lot more patients that they can have access, and hopefully one day it becomes a point of care, and this therapy can be in a lot of medical facilities all over. So
wonderful, great. And then we'll hopefully talk a little bit more about reducing it from 24 hours, just saying. So Celia, one of our key themes, as I just mentioned, was expediting the transfer of from research all the way through the clinic and commercial. I'm wondering if you can elaborate on selected stem cell programs at the Broad Center for Regenerative Medicine at UCLA. And I'm wondering if process intensification or optimization is a key strategy for you there.
It absolutely is, although I have to admit, I had to look up process intensification.
Okay,
so
that's okay, we're all friends.
we don't know that that's what we're working on, but we definitely hear that that's what we need to do. One of the lead programs that I've been here talking about in partnering meetings is a 9 to 12 month process. And after I tell partners that, I usually have to help them wheel their chin off the floor. So that's cell therapy for a range of blinding eye diseases. It's a RPE replacement therapy. It's not an oncology application, so we do have more time. But it's still not feasible to really imagine doing that commercially. There are a couple other projects behind that in terms of delivering muscle progenitors, some liver replacement. There's a lot of interest in various neurological applications, but all of these are long processes that take a lot of time for the cells to mature to the point where we would actually imagine delivering them. So we think a lot about how in the academic setting to make the right investments, that these projects are going to be partnerable, because most potential partners, even the startups that would spin out, would never be able to take something that's that clunky fully to commercialization. So we think about helping these programs by focusing on assay development so that as we're improving the process all the way through, we can still be sure we're hitting the same targets. That wasn't something that academics necessarily thought about before, but if we can support these programs with that kind of development, they become much more amenable to continued development. We're also trying to think about, what are the common touch points and can we get those working well? Everything passes through a pluripotent stem cell, say, at least in the work that my group supports. Is there anything around that we can intensify, streamline, solidify way upstream, much earlier than an academic ever would have imagined doing it, so that we can get this moving forward and moving forward faster.
So it's a great segue actually to Uma, who does quite a bit of assay development, but I'm wondering if, continuing from Celia's description of how she's translating science to therapies, can you talk a little bit about the process development work that you're doing at Patheon Services to support the industry?
Absolutely. So I lead the translational services, which is almost a step before process development. So a lot of times if a process is not designed right, then you're trying to optimize a suboptimal process. So what we do in our facility is to really look at fit for purposes unit operation and put them together so that you're designing with the end in mind. And again, as Betty said, a lot of our customers are academicians who are moving at the speed of light, and so there's a need to kind of slow down and look at the quality aspects of it. Can it be commercialized in the long run? So and again, because cell therapy by nature is bespoke, we can't just start from scratch for everything. So what we've developed is for key workflows for iPSCs and T cells and for analytics and things like that, we have something called a rapid development framework, where we know the operating edge of each unit operation that really can be kind of stringed together to build a workflow that hits the requirement. So yeah, absolutely. I think the new word that we use, the process optimization, it's even before process development, because you got to kind of choose the right unit ops before you can kind of call it a process.
Wonderful, thanks. So I'm going to start opening up questions, so anyone who wants to answer, please feel free. Karen, I am going to start, though, with the ingenuity process, sorry. So one of the unique challenges, I think, with your process is the diversity of incoming patient-derived material, right? How are you mitigating risk tied to that for your accelerated process?
So actually, in the field of autoimmune disease, we see much less variability across the different patient indications and prior treatments and all of the rest. It's a more standard starting material than the oncology field has encountered. And the quantity of different types of cells are more in the normal range. So we really have not had that issue. We have looked at T cell yields across the different patient populations and different prior treatments and have not seen that be a challenge.
Well, lucky you, right?
Exactly.
So maybe I will redirect to your neighbor there, Saba. I know that you do experience some challenges associated with that diversity in the input material.
So, I just want to stress that out again, that overall, throughout, all these processes, even the, we might have sometimes trouble with the standard, process 9 days that, we try to meet the dose and we can't. There are patients that we can't do. In fact, I think one of the early patients, one of our very successful stories that is always in the news, we never met the dose for that patient, and we just,
Well, you could.
We just go ahead,
Yeah.
So the thought, again, I think we really, really have to stress is that we don't need a lot of cells. We just need the right cell, right? I think quality of the cells are way more important than quantity. And if we have the right cells, we can work with one cell that can, cure. So this is, this is, I think, like, the change of thought process that we have to have, that we do not need to generate large amount of cells. We just have to generate the right cell so that, we get to efficacy of the trial, so.
And I don't know if we'll get there today, but analytics play a big role in that as well, right?
That's correct.
Okay, so maybe I'll continue with Saba. Again, feel free to jump in, but part of the process optimization, we heard already about eliminating leukapheresis is one way of getting at that shortened time frame, but also the obvious step with cell expansion is an area that we've been focused on. So just wondering if you all can talk about that particular step, as well as potentially other steps in the process that we may want to target to reduce the overall manufacturing or innovating time.
Sure. So, in order to optimize the process, or now we're focusing on shortening the whole process, specifically for cell therapy, it's a very complex process, right? We have from the cell selection, activation, transduction, expansion, freezing, testing, right? There are like all this step, and if you look at it as let's say we say a step from 1 to 10, right? Some areas like expansion that we've been doing very successfully to shorten it, right? It's easy. It is easy. I don't want to say it's hard. It is easy to shorten the process, but there are, if you look at to shorten the overall process, we have to really look at every single step and see how can we optimize each step, right? Maybe the transduction time is not 14 days, right? But if here, like in one step, we can shorten it for half a day, and then the other step we can shorten it for another half a day, and overall, I think we can optimize the process. And for process optimization, it is really essential to look at every step. For example, the transduction time, right? I know again, like the Thermo, the DynaSelect that can select and activate at the same time, right? That's a great, saving the time compared to the standard process, which is, we select, we activate, wait one day, and then introduce the lentivirus and then expand, right? That's one space. And the other thing is, it's not necessarily, the time that we have to change in order to shorten the time, right? And I tell you what I mean. Sorry. So, for example, think about transduction time, right? If you look at the lentivirus, let's say we try to develop better lentivirus with higher quality, so that we can get into the CAR percentage, right, that we want in a shorter time. That's one step. That's the work that we can focus on to see how we can shorten the transduction time, or the media that we use. Can we design a custom design media that improve the transduction so that we can shorten the transduction time? So these are, or the bioreactor that we use. Can we use a design that improve the transduction efficiency, which I know a lot of work has been done that stress about the geometry of the bioreactor. So necessarily what I'm trying to say is that for every process we shouldn't say, oh, can we do it this process from 3 days to 1 day. We have to look at every element to see how can we change it so that we can reduce the time, reduce all the extra step, or reduce the cost of goods. So all of this stuff, with having in mind that we don't compromise the potency and of course, like safety and the standard of having a good or potent cells, basically.
So can I add to that? So I agree. I also think we need better analytical tools. One of the things that we need to think about is how can we measure dose before protein is expressed? And how can we measure purity of the product and what purity matters? But I'm talking more about process-related impurities, that they are quantified and that we know how they are going to contribute to the final product, because you don't have time or volume exchanges to dilute out. And everybody knows that these processes don't have viral clearance steps. So while for an autologous process that's less of a concern, for an allogeneic process that's a big concern. So we really need to understand all of those different attributes of the product. I'll say another thing, that on the QC side, the number of cells we have to take for our QC analysis is 3 times the therapeutic dose. That seems counterintuitive, so we should be able to manage that better. And from a time perspective, the QC time is still 2 weeks. And so we need to look at that. Yeah. And it's no longer sterility that's the problem. We've gotten that down. It's the potency and other quality attributes of the product itself. So I think we need to focus on those as well.
So next year's panel on analytical improvements. Celia, I don't know if you had anything to add.
I was going to chime in for the analytics. You know, a lot of the cell types we're working with are adherent, and so if we want to sample, we destroy. So how can we be doing a better job using image-guided, understanding what we're learning from the images in process? You know, that takes a while to develop, but it will let us know much earlier if a culture is on the right track, and we are dealing with autologous products. Or if we can shorten timeframes, because we've now know that culture is committed to where we want it to go, and we can prove that. So that's something that we're working hard on.
Great.
So yeah, to follow up on that, adherent cultures, especially iPSCs, what we've started doing is implementing AI-based image analysis so that you are not relying on an operator to make the call. So definitely more use of AI. And coming back to like, how can you minimize the number of cells? I think it's like you trying to develop assays which are multi-attribute, right? Like you can measure multiple things at the same time. And I know it sounds like a unicorn right now, but I think there are enough technologies out there which over the few years, I'm pretty sure that can be leveraged.
Yeah, I agree completely. So maybe I'll jump to another mechanism for optimizing the process, or at least making it more efficient, and that's automation. So this was a theme, I think, in one of the workshops yesterday, and all the workshops, I have to say. But what role does automation really play in your optimization of your process in your respective facilities, whether that be in part, certainly less likely that it's going to be in whole at this stage of the game?
So maybe I'll start, but everybody else, please chime in. We have a huge interest in automation, especially with our shorter process, because if we're going to go with a very distributed model, that's going to be an expensive footprint to maintain unless you can reduce the climate controls and cleanliness controls for the facilities, and also the labor costs. So we would really be interested in full automation and fully closed systems and at-line analytics. Let's just be clear. And I think then it's really going to improve reliability and feasibility of having a very distributed model, which gives access to lots of patients. That being said, it's got to be the right automation. I think one of the challenges of early attempts to automate these processes was that we put them on platforms a bit too early in the innovation curve on this field, and you couldn't it constrained you in your ability to innovate. And so I think it is really important for innovators and technology innovation to come together and jointly address the problem statements.
Good, really good feedback. Thank you.
I can say a little bit about from a manufacturing perspective. So definitely the biggest driver for cell therapy is reducing cost, right? And so especially with the 3-day and 1-day process, the amount of and then we have a bigger addressable patient population, it's absolutely important to have automation to serve the demand and also keep the costs low. So I agree with Karen completely that manufacturing facilities in partnership with technology developers and therapy developers, I think there's a mandate to kind of find solutions to address the current problem. And so yeah, we are very interested in that as well.
I agree with both of you, but I have to say that automation is great, right? It standardizes the let's say the manufacturing, but it has to be close to the commercialization, not early developer, right? Because as you mentioned, one of the problem of automation is the adaptation. You start to work with a process, and you want to be able to manipulate, to change, and it's at least the cell therapy is such a fast-paced field, right? Constantly things are changing, and so you can't automate your process in the beginning, and then you say, oh, I spent this amount of money, right? Because it's very costly as well, and now we can't change it, right? So I think it's very important. I think it's very, very important to first simplify the process, optimize the process, shorten the process as much as you can, right? Because it's very hard to automate like a 10-step process or like 2 weeks process or a month process rather than automating like a 1-day process, right? Hugely different in terms of cost, complexity, right? Once you develop your process and you're done and it's almost ready for commercialization, yes, I think automation would help, of course, with the labor, with the standardization, and there are a lot of benefit. But right in the beginning, think of automation. Everyone has to be careful. Otherwise, you get stuck to a situation that you don't know what to do. So,
I see it a little differently because I have definitely had potential partners tell me, don't mess with that. You're going to make choices for us that we don't want you making. You're going to constrain us. But at the same time, for these programs that I'm thinking about, if we don't do some work on it, then they will never go anywhere. We really do need to show that at least certain components can be automated, can really be made scalable. And for the ones that I'm thinking about, automation is the key to scale. So especially, because we're not going to larger and larger volumes, we're going to more and more different patients for scale. So we're really trying to figure out what is that sweet spot, what is the right, for us it's going to be focusing on certain unit operations that we can say, okay, we've done this, they're amenable to straightforward automation plays, you can take it from here, but we've at least done some work and some thinking about it.
That's correct, right? I think maybe a solution to that is that we hear GMP in a box, right? From start to finish, automated. Maybe we break that down, right? Yeah. Especially if that GMP in a box takes 3 weeks, right? We don't want to automate the whole process, and then we give ourselves, and then they are on, they have to pass several steps. Maybe if we make it more like in a unit operation, and then automate a specific unit that in any case we need to automate that, that side of it. So that would be a way.
I think we're all in violent agreement. When I talk about automation, I'm not talking about putting it all in a box. That is where I think the industry maybe took a left turn early on. I think what I'm talking about is modular automation and interaction with robots that can do the manipulations with the instruments that you use as the product and process developer to make the process automated, and they are programmable so they can do different steps in the process. That's, I think, where the field will really mature. And I know that's potentially controversial, but I think where we are as a field in our maturation, that's really what we need, because cell therapy is such a broad field, and there's so many different types of processes and cell types and products that putting all of these in a box is very difficult.
Okay, so you're leading into the next questions, which is great. So modularity and flexibility are really where we focused a lot. Years ago, I'll say, do you feel that the industry has made efforts and progress in kind of moving more towards standardized approaches, platforms, or do you still think we're heavily in the modular, has to be flexible to accommodate some of the challenges that you just mentioned, Karen? So just curious what the consensus is within the panel now around modular versus all-in-one.
Anecdotally, UCLA's GMP is in the process of trying to move towards centralized protocols for certain what they thought were consensus steps. And I'm hearing the PIs complain nonstop about how that's constraining their flexibility, because they're really in the R&D getting into the process steps and wanting to be completely bespoke. So that is still a very dynamic tension that's very active. But I will say that from last year's meeting on the Mesa to this year, I've also seen enormous progress in terms of people moving away from sort of all-in-one concepts and trying to tackle processes start to finish in one box, and instead being very flexible in stringing together beads on a string to get us to the improvements that we're looking for. So that's been really exciting to just see 12 months worth of progress in that regard.
Agree.
One thing I'll add is that, so you can't really, like, if you take something that are multiple devices that does the same thing, right? You can't just say this is the best and everything else is bad. I mean, we are very agnostic, and sometimes some instruments are best for a certain process. What we try to instead look at is, can you standardize it? And is there a way, like I'll take the example of cell counters. There are multiple versions of cell counters, and what we do to standardize is we use the NIST-based, there's an ISO counting method, and so we implement it for every process to make sure that whatever counting method or instrument is used is valid for that workflow. So I think a combination of that is a better approach rather than kind of being very prescriptive about what to choose and what not to choose.
Okay, so kind of moving on to bringing in new products into your process and new innovations that are happening all of the time. How do you balance between the desire to bring the newest, the shiniest new object, I'll use that a little flippantly, but to bring in new technology into your process while minimizing the risk? So just wondering if what's your strategy, your approaches for that?
So I think I'll go back to something that Scott Gottlieb said yesterday, which is my hope is that at one point we will understand our products and our processes sufficiently well that we can ease the journey to introduction of change. You notice I'm not talking about analytical comparability or well-characterized biologics, because cell therapies will never be that. But if we can understand what makes the product beneficial to the patient, what's important to control, and that we demonstrate that level of control in the face of change, I think that that will be success. And I would like to say, this is a pipe dream really, but we could maybe consider it our North Star, that we can have adaptability or agility built into our processes to be able to make the right product for the right patient each time, and not be married to the dogma that the process is the product, because I really do believe in cell therapy, the product is the product, the process is the process, and we understand each process step and what it's intended to do sufficiently well that we know the impact of changing it to the product.
Well said.
I think there's a difference between being in academia and industry. I think in academia it's a little bit easier. We're more flexible to try new things, compared to industry, because industry you probably come up with a process and you have to develop that process and then stick to it. But at least at Penn, we constantly monitor new technologies and test them, and actually we have a unit for that just go around and see what's out there, what's especially now that there are a lot of new technologies in terms of real-time measurement of quality of T cells, like fast sterility tests, PCR-based assays, like that is different than what has been out there, and see can we use it. So we have a little bit of flexibility in that, but of course it's always hard to just implement it and integrate it into your process. I think that goes back to that automation, is that we have to when we start thinking of a process, we have to leave room for adaptation to new technologies, to new processes, to see what if now we can measure glucose at real time at every second that we want, right? Which we know that we can right now. So does that help us in the whole process design and process development? So I think it's very important to be constantly in touch and then see new technologies and development out there. So
I have a different perspective from a service industry. I want all the shiny new toys. But the problem is it adds to the operational costs, right? So you constantly have to balance what is really truly required. Is there a sufficient demand, or is it serving a critical gap? And once that assessment is done, that's when we usually bring on. But it's hard to bring everything that's out there.
So in terms of technology and innovation, I'm wondering if you could share within your own facilities what you have perhaps evaluated and implemented that's really moving the needle in terms of efficiency or reliability in your process.
You stumped us.
I can say like some, for example, we work with different bioreactors, and that in cases that help us to pick for different processes. Actually, we use different bioreactors, and those are I would say like new development that we're using in our processes. Or we right now switch to a lot shorter, to a few hours of sterility testing. It took some time for the decision because of the cost, because of the new training, and because of the space. Because if you think about it, it's like having a kitchen, and then you want to keep going with a new coffee machine or new fryer or new. So at some point you lose space. So we don't have literally space to just even try all these processes. But there are like certain elements that we know that they are really sub-optimized. So we try them, and then we have new sterility testing. Now we're working with Thermo for using the optimized process for cell selection and activation at the same time. So I think these are all, each of these elements for our process that would help, it helps in terms of optimization and moving forward with the new technologies and the benefit that they will bring in terms of getting better T cells and faster T cells.
I think for us, really the advances in analytics has been a huge accelerator. The imaging technology, the single cell analysis, the more precise molecular biological methods, those are all really important in our being able to understand the cells and what's happening. So I think they're really, that's the accelerator, even though I want to accelerate more.
Maybe I'll rephrase a little bit here. Is there a technology that you are really excited about that you think is going to make a difference in your manufacturing process in the next, I'll say, 2 to 3 years? So, again, I'm not getting you to name names or get real specific, but just in general, technologies that may excite you. You mentioned single cell analytics, or it could be certain bioreactor technology, but just trying to kind of get a grasp for the audience on what we should really be looking for from a technology horizon point of view.
I would say something that I'm very excited is looking at the metabolic properties of cells and use that as a measure of the quality of cells that you're working with. And now we know that they're like, for example, we use CyTOF, the LCMS, to just measure all the properties, which is very complicated. I always have to have a team explaining all these metabolic properties and relation, but that's something that can be implemented. And instead of maybe the current standard that we only use flow cytometer for some cell surface marker, but that's something that can add it to the process as a real-time measurement of the quality of the cells. So I would be excited to use that in our processes.
I kind of agree. We see a lot of requests for metabolomics and lipidomics, and when we start collecting the data trend, it kind of like tells you a lot more than just a viability or cell health would say. So that's definitely advanced analytics in general of any sort that gives you a window of more than what you're looking at right now. And in addition to that, automation and AI, I think it's really exciting because it's not just for process, even for analytics. If you can automate a lot of processes, it takes away manual errors, fail rate, it increases the speed in verifying the assays and stuff like that. So in general, I think automation and AI, if leveraged completely at different parts, which are current bottlenecks, could be a huge benefit.
And I'm finally seeing automation interact with 2D cultures, which I'm very excited about. So I've been asking for that. I will keep asking for that. I'm putting it out there to this room. But there's more and more of that coming. And my sense is that when people were thinking about automation in terms of robotics, it originally was extremely cumbersome to automate a line, and that's becoming more and more flexible and more something that we could imagine actually putting in academic labs and being able to flow that through to continued development. So I'm excited to see those two things continue to mature.
I think lastly, the data, the ability to real time assess the data across the whole value chain and understand what attributes of the product lead to clinical outcomes and how to adjust those so that you're providing the best outcome for patients. I think this started as being a real need when the first CAR therapy products were under development. And certainly when the gene therapies were under development, and now there's an industry that is starting to be able to allow that broad insight, and that's really very, I think, motivational.
And it's obviously not unique to cell and gene therapy, but I would say that that holy grail has been an ambition for pretty much all the biologics and even small molecules, to be honest.
But I think it's a unique, particularly the autologous therapies, whether it's a bespoke gene therapy or an autologous cell therapy, because each batch is a patient. You can derive a different level of insight than with a batch that serves multiple patients, because you have that variability of patient response that it's difficult to tease apart. Is that a process thing you could tune, or is that a patient-specific attribute? But if you've got a single patient, single batch relationship, it validates a complete response. Whatever the condition of that product was, it can lead to a complete response.
Okay, so less than 10 minutes left. I think that one of the unintentional themes, or maybe it was intentional, that also came out was the importance of collaboration, and this ecosystem that we all operate in only works if we do that effectively. We talked about some of potential collaborative engagement that it could include automation, engineers, and technology providers, and I think really the list goes on from there. But wondering if you can talk about collaboration, whether that be within your organization, whether that be between academic institutions or with industry or with regulatory bodies or with funding, CERM to be specific. Wondering if you can talk a little bit about how the collaborations have impacted your goals and objectives, hopefully for the positive. So I'll start with Uma.
Yeah, I mean, for us, I think it takes a village, right? So even within Thermo Fisher, I think the ecosystem that we have, the reason why we can be kind of successful in the services part of the business is the vast expertise that lies within the company, be it like, in product development in terms of equipment or an analytical assay or whatever you are looking for. So just that ready access has been really helpful, because then you're not second guessing yourself or doing something that's not the best method to do. The second thing is that working with our customers who are coming in, because they have very unique needs, right? So trying to understand, not imposing our stuff, but instead kind of like adding our expertise to what they're already bringing to the table. And then beyond that, it's standardization bodies. We work very closely with CERM, we work closely with NIST, and international organizations like ISCT. I know Tony is here, I have to give a plug for ISCT, ISSCR, and so those organizations, because each part of it brings a unique perspective, and I don't know how you could work or operate otherwise if you're not completely embedded in the ecosystem.
Good answer.
I also think collaboration is absolutely essential. We can't do it all ourselves, right? So I think, in our case from academia, we have to collaborate with industry, with regulatory compliance. So to be in early stages, if we're trying to develop, a therapy or develop a process, we have to use industry expertise. We have to, use like academic flexibility to try new things and be in touch with, all this regulatory, and to make sure that we have the highest standard of, of regulatory processes and safety for the patient, and also, with funding agencies, right? So we have to really work together, and so that we don't reinvent the wheel when there is, let's say, like an industry that is really an expert into some processes, and we have to work together early on. Before we start, an early developer, we have to be in touch. We have to be constantly, I think, in communication in order to have an optimized process and and move fast in this field, in such a field that, it's like really fast paced. So very, very important. We're actually true believer of teamwork makes the dream work. Otherwise, it doesn't work, right?
Yeah, I don't know if I have much to add. I think that collaboration in all forms is critical. Certainly between the research institutions and industry, we should lean into each other's strengths. We should not try to compete with each other. I think that there's huge value in having collaborations where you can get deep scientific understanding that helps advance the field by bringing teams together and working to bring new insight and new technology to play. I think also within a company, breaking down the functional silos and working as a truly integrated team is vital to bringing these products to patients. And then, of course, not just FDA, but we want to bring these products to patients globally. So working with WHO, working with EMA, working with all of the regulatory bodies around the world where you have patients who could benefit from your products, this is really important. And then the technology that allows us to bring those in a cost-effective way to those patients. It's really a global economy.
I get to live it in a really interesting way because I'm at a University of California institution, so we have sister campuses. We're within California with all of the support of CERM and the sort of ecosystem they have very much helped build. And then even within our campus, I see one program seeding the next. It's been fantastic the way sort of the pioneers on campus have turned around and said, don't reinvent the wheel. There's too much to be done. Here are my protocols. Here's what we've learned. Here's the nitty gritty of how we've had great partners in industry and again, helping us get onto platforms faster, helping us optimize so that we're not spinning wheels. Everybody just feels like we've got so much to do. Let's not repeat anything. And so I've really seen that play out in fantastic ways for the cell therapies that are moving forward. It's been great.
Terrific. Well, I think we're pretty much at time, but I wanted to say thank you to the wonderful panel of females that are all up here anyway, and I thank all of you for coming out. I don't know whether we have time for maybe one or two questions from the audience. I think we technically have 2 minutes left. Any? Yes, please.
One question. My name is Bill Mercer. I live my life in apheresis, and it's great to hear that alternate sources of the whole blood are being considered. Question. Whole blood has a lot of contaminated cells. To what degree of specificity, to what degree of purity do you need? And the second part of the question, is there a jewel lymphocyte in there that if you could just get that one cell, that would even solve more problems?
So the question for those of you who may not have been able to hear is switching from leukapheresis to whole blood, of course, increases the number of impurities or cellular impurities that are in the starting material. And to what degree is that a problem? And then the other is there a specific subset of the lymphocytes that if you could isolate it would lead to better outcomes? For us, because of the Thermo technology to isolate and activate at the same time, we actually do get very pure T cells out of the whole blood, and so it's not a problem. We have very high purity, similar to isolation from leukapheresis, and we have the data to show that. Within the T cells themselves, That's a more difficult question, and maybe I will have Saba address that.
So it's a big question. It's a big question that what's the best subset and what's the best T cells? The only thing I can say towards that is that I am very sure in the very near future we would have designed target for cell therapy that, maybe, what exactly the T cell subsets and what's the optimized process around that could give us the best response based on the knowledge that is out there.
Okay, I think with that, we'll close the session. Again, thank you for coming, and thank you to our panelists.
Thank you.